Pasamos a la última comunicación, ACE Virus Suppression following AAV-mediated delivery of closer to germline monoclonal antibodies in rhesus macaques. Presenta el Dr. Martínez.
Good afternoon, everyone. I would like to thank the organizers for the opportunity to be here today. And as you can see, I'll be speaking about AAV-mediated delivery of closer to germline antibodies in rhesus macaques. AAV, or adeno-associated virus, is a small virus which infects humans and other vertebrates. It's not currently known to cause any disease and is very efficient at infecting and delivering, their genome. So it's currently being exploited for gene therapy. Basically, we remove the whole genome and we put our gene of interest, and then it gets encapsulated, and basically you can inoculate it intramuscularly. And now the muscle cells will become factories of the antibody you put in the AAV, and then you can basically produce antibodies in your own body. Hopefully those antibodies will neutralize HIV. This, of course, circumvents the difficulties associated with generating a successful immunogen or vaccine. We can use well-characterized, broadly and potently neutralizing antibodies. And there is no need to produce those antibodies in large amount, and of course you don't need several administrations of those antibodies. So technically, one single administration could account for years of antibody expression. Therefore, we think the delivery of potent and broadly neutralizing antibodies with AAV vectors is a promising approach, and it would actually work well for both approaches for prevention and treatment of HIV infection. Today, I will be focusing on the therapeutic approach, and as many of you know, we have one rhesus macaque that in the chronic phase of the infection of a SHIV-88 infection, which is a simian human immunodeficiency virus, a very robust model that generates very nice viral loads. We administered AAV encoding for a cocktail of antibodies, and this monkey has been suppressed ever since. As you can see, all the viral loads were undetectable, and this is over 6.5 years now. We have two additional macaques that receiving also a cocktail of antibodies with AAV were suppressed for over 3 years now. However, this is not always the case, and as I will be showing here, this monkey, which has been nicknamed in the field as the Miami monkey in analogy to the Berlin patient, had very high levels of 3BNC, which is a CD4 binding site antibody. However, the other 3 monkeys inoculated with the AAV in the same cohort did not have detectable 3BNC, as you can see after some time, and that was due to the immune responses generated to 3BNC, what is called ADA or anti-drug antibodies. As you can see, the Miami monkey in red was the one with the lowest ADAs, and that's why we could see that antibody. Similarly, the case for the other antibody that the monkey received, which is 101074, which is a V3 loop antibody. And in this case, we had 2 monkeys, and in this case, the Miami monkey was the one with the highest levels. And as you can see, that correlated nicely with the ADAs. Perhaps a more extreme case was the third antibody that the monkey received, which is 10E8, which is a GP41 antibody, an MPER antibody. And as you can see, all the monkeys mounted that really a strong immune response to the antibody. So the system not always works. We characterize in detail the ADA responses, and what we found out is these ADAs mostly target the variable domain of the antibody, so they are indeed anti-idiotypic, which is not good news, because you can change all the parts of the antibody, but you cannot really change the variable domain, because it's what is binding the antigen. Also, we saw reactivity to the CDR3 in the heavy chains, which is the most important and the one that really is the one predominantly selecting the antibody for its target. Importantly, for the talk today, point number 3, the levels of ADAs correlated well with the level of antibody mutation. So once we delivered several antibodies in a number of monkeys, we realized that there was a really strong correlation. So the more mutated the antibodies are, the more divergent from the germline, the more immunogenic, which makes sense. The more they differentiate from your sequence in your body, the more foreign for you. Then, as you know, all these antibodies, which are broadly and potently neutralizing, are generated in only a number of individuals that live with HIV, and it takes years. So they undergo an extreme process of hypermutation, and this is the percentage of identity with the germlines of just like a handful of PNAPs, of broadly neutralizing antibodies. And what we realize is that there are some which are naturally more self. They are less hypermutated, so the percentage of identity with the germline is higher than we thought. Our hypothesis was what would happen if we delivered those antibodies. In this case, after some screening and testing, we selected these three, which are DH270, which is also a V3 antibody, PCI-N63, which is a CD4 binding site antibody, and DH511, which is also an EMPER. This is a similar combination that received the Miami monkey and the other monkeys in that cohort, and the rationale is to avoid the virus generating escape mutants, or to make it difficult for the virus to mutate. In this case, we did a 4-monkey experiment as well, and these are the viral loads, and about at week 14, at week 14, the monkeys received AAV encoding for these 3 antibodies that we mentioned. And actually, when I submitted the communication, we had 2 monkeys that were nicely suppressed, which is 50% of them, which is the best percentage we had ever had. But recently, the last 2 viral loads in one of the monkeys show a rebound. The 2 monkeys that were suppressed, this one and this one, are suppressed because they have nice levels of 2 of the antibodies. In this case, there's the V3 antibody and the CD4 binding site antibody. And the 2 monkeys that are not suppressed, we have 2 different cases. We have this monkey that has really high levels of 2 of the antibodies and even really detectable levels of the 3rd one, but the virus is just growing and swimming in the antibody. So we speculate that this monkey had an escape mutant to begin with. It's also the one that had the highest viral load, so it would make sense. And then we had the other monkey that was not suppressed, which indeed didn't have much antibody delivered, and that correlated with a strong immune response to the antibodies. So basically, when we compare now the data that we obtained with these antibodies that are less hypermutated, we see that 3 out of 4 monkeys had really good levels, while before, with the same type of antibody, a CD4 binding site antibody, only 1 had really good levels. And on the other hand, with V3, we have a similar result. So now we have 4 of 4 monkeys with detectable antibody, when before we had only 2. And even with the most difficult antibody, which is the EMPER antibody, we have now 2 monkeys with detectable antibodies, when before we had very little levels. So my conclusions for today is that the use of closer to germline antibodies may be a viable strategy for avoiding the ADAs, following gene therapy with this approach of AAV and antibody vectors. Our data supports the potential for long-term suppression of viral loads with this approach, with the AAV antibody approach, but it also highlights the difficulties associated with achieving such long-term suppression, meaning that the virus may be still able to escape in some conditions, so we have to think about using maybe a 4th antibody or finding a really good combination that makes it really difficult for the virus to escape. Then, of course, eradicating or minimizing these ADA responses would be crucial to make the AAV delivery of antibodies a consistent and reliable approach against HIV. And then these are my acknowledgements. Of course, the lab, including Don Rogers, which was my mentor, Wan Ping Gao, which made the AAVs, Jeff Lifson at Frederick, which made the viral loads, and of course the Wisconsin Primate Center people, which took really good care of our monkeys, and the funding bodies. And thank you very much for your attention, and I'll take any question.
Muchas gracias. ¿Preguntas? Muy bien. Muy gracias.
A vosotros, gracias.